Hi guys, welcome back to my channel. I'm so glad to see you all here. So today we are going to discuss a bioinformatics file format that is very commonly used while performing variant calling analysis, and it's called variant calling format, that is VCF. So you all might have come across a VCF file at some point, and today I intend to discuss how this file is generated, more importantly, what information is stored in this file and how the information is organized in this file format.
A variant call format file, that is a VCF file, is the output of a bioinformatics pipeline. So basically, this is a text file that stores gene sequence variations. So in a typical variant calling workflow, a DNA sample is sequenced through a next generation sequencing system and produces raw reads. These reads are stored in a file having format FASTA or FASTQ, and these raw reads are then aligned, which gives us the BAM or the SAM file. We have previously discussed all these file formats, so if you are new and if you haven't heard of these file formats, and if you would like to learn how the data is organized in these files, then I recommend you to watch those videos. I will link those videos in the description section below. Now, using the SAM or the BAM file, a variant calling algorithm or tool identifies changes in a particular genome as compared to the reference genome, and the changes are then recorded in a variant calling file or a VCF file. So sequence variations like SNPs, insertions, deletions, structural variants are all recorded in a VCF file. Some VCF files could be very large, so in such a case it would not be advisable to load the entire VCF file in memory. In such cases, different strategies could be used, like using Linux or Unix tools to manipulate or open only a part of the file or only fetch the relevant information from a VCF file. A word of caution: a VCF file, however, is a text file, should never be opened in a text editor like Word, Microsoft Word, as this could screw up the format and could corrupt the files. So, such a file should always be opened in Linux or Unix environment, and it's safe to open such files in such an environment and take a look at those files, or even subset few lines, or fetch relevant information. But such a file should never be opened in a word processor like Microsoft Word. So when you open a VCF file, essentially you should be seeing something like this. So these are the first few lines of a VCF file, and again, a VCF file could be divided into various sections. So basically, a VCF file contains three sections. The first is metadata lines, and these are multiple lines. These are the first few lines with which the VCF file begins, and these files are prefixed by £2 symbol. The next section is the header section, which is basically a header line, and this line is prefixed by £1 symbol. And followed by that, we have records. These are data lines, and there is various information that is stored about the variants. And we will be going into the details of each of these sections in the following slides. So starting with metadata lines, these are the first few lines in a VCF file, and these files describe the format and content of a specific VCF file. So these lines could include information about sequencing performed, the variant calling software, or the reference genome used for determining variants. As I mentioned previously, these are metadata lines and they start with £2 symbol. You will see all these lines are prefixed with £2 symbols, and the first line here is the file format, which specifies what format is this file. Followed by that, we have various other lines with a bunch of information. And followed by that, we have a section of lines that start with info. We have few lines starting with filter, and we followed by that we have lines that are starting with format. So basically, the line starting with info has some description of the annotations that will be used to describe the variance sites that will be found in our data set. So for example, in the data lines for each variance site, there will be some information on the number of samples with data, the total depth, the allele frequency, and so on and so forth. So for all those annotations, the description and the expected value for those parameters can be found in the metadata lines starting with info. Followed by that, we have lines that are starting with filter, and this basically tells us what filters have been applied to our data. And lastly, we have lines that are starting with format, and this is similar to the info lines. So basically, to describe a sample, we will be having information in the data lines for each sample, their genotype, the genotype quality, the read depth. All of those informations will be present as annotations and a value, and the descriptions of the annotations and the values expected, the format of the values, are described in the metadata section with the lines starting with format. It's okay if it's not very clear as to what the lines starting with info, filter, and format mean. Hopefully, it will be clear when we move on to talking about the records in the VCF file, talking about the variants and the information that is present for the variants as well as for the samples. And at that point, we will be talking about a lot of these annotations. So, hopefully, it will be clear by that point. Now, coming to the next section, which starts with a header line, and the first 8 columns in the following section are mandatory columns. These are tab delimited columns, and they represent the properties observed at the level of variance. However, if there are multiple samples present in a VCF file, then some of the site level information will be average or summary of the values obtained for that site from different samples. Followed by that, we can obtain sample specific information such as genotype, genotype quality, in a column called format, which is the ninth column. And followed by that, we have individual columns, one column for one sample if there are various samples, that provides annotation values for each sample. So those will be sample column 10 and beyond. So if there are multiple samples, then each sample will have its one separate column. The lines following the header line are the record lines. So basically, these lines store information about the variants and various information about the variants as well as about the sample. So let us go into the details of each of these columns and take a look at what information is stored in each of these columns. So the first column is chromosome, and it holds information on of what chromosome was the variant found. The next column is position, so it basically provides a position of the variant in that chromosome. If it's a multi-base variant, then the position corresponds to the first base's position. If it's a deletion event, then the position corresponds to the base preceding the event, that is before the deletion. The ID is basically to indicate whether this variant has previously been classified or and present in any database like dbSNP. So based on the variant, their position on the chromosome and the location, whether this variant has been previously classified and is it a part of a database. If it is, then that identifier is present here, so that will help us identify these variants across many samples. If not, then a dot indicates that this variant has not previously been classified. The next column is reference. So basically, at that position in that chromosome, what is the base that is present in the reference genome? And the alternate is basically what base is present in our dataset at that position in that chromosome. So basically, the alt column holds the variants that are found in our dataset. In case if there are multiple variants present, then they will be comma separated. And for events like insertions, the alt column will also include the bases that are preceding the insertion. So one could understand where the insertion is compared to the reference sequence. And just a quick note that these bases in the alt and the reference column that are reported are always given on the forward strand. So the column following the reference and alternate allele is quality, and the values here are probability values estimating whether the variant called at that site is due to sequencing error. So basically, a value of 10 indicates that there is 1 in 10 chance that the base that is called is actually not a variant, but it's due to sequencing error. So basically, a higher quality indicates that we are more confident that this is a variant and not an erroneously called base due to some sequencing error. So the next column is filter. So basically, there are some filters that we apply to our dataset, and if you want to know what filters those are, you refer back to the metadata section to the lines starting with filter. There you will be able to find the description of what filters were applied to the dataset. So in our example, there were 2 filters applied. One of them was to detect variants having a quality score less than 10. So here in this example, the second record seems to be failing that filter because the quality for that variant is 3, which is less than 10. So that record is flagged as Q10, which is the name of the filter. So all the other variants seem to pass that filter and hence has value pass. If there is no filtration applied, then for the records, then you will be finding the filter column having dots in places of pass or the name of the filters. The column following the filter column is the info, and here you will find information for certain abbreviations for these sites. And if you want to know the details of these, the description of these abbreviations, then you can refer to the metadata section and refer to the line starting with info, where you will be able to find the description and the names of these abbreviations. So here we see certain abbreviations like NS, which stands for the number of samples with data, DP, which is the unfiltered combined depth across all samples, and AF, which is allele frequency for alternate alleles. I will be talking about allele frequency and how to calculate that in the upcoming slides. And again, we see additional abbreviations like AA, DB, and all of these abbreviations and the description can be found extensively on GATK forum, and you'll be able to find documentation for these abbreviations and what values they expect and how to interpret basically these values. Now, finally, moving on to the sample level annotations and starting with the column format. The format holds a colon separated list of abbreviated categories, and these are descriptive of the samples. So, if you want to know what each of these abbreviations mean, again, you can refer to the metadata section to the line starting with the format to get an idea on what these abbreviations stand for and what values do they expect. So the colon separated list here usually begins with GT, that is genotype, and here we have genotype followed by genotype quality followed by depth and followed by haplotype quality. So to give you an example, look at the last record in the format column. For the last record, the format column has genotype, genotype quality and depth. So the data for the following samples will follow the same order. So the genotype for all three samples are, so for sample 1, the genotype is 0/1, for sample 2, the genotype is 0/2, and for sample 3, the genotype is 1/1. Followed by that, we have genotype quality. So genotype quality for sample 1 is 35, genotype quality for sample 2 is 17, and for sample 3 is 40. And lastly, the depth for sample 1 is 4, for sample 2 is 2, and for sample 3 is 3. In the following slides, we will be looking at what the genotype is, what does 0/1, 0/2 and 1/1 mean, and what is the difference between the slash and the pipe operator. So we will be understanding all of those details in the upcoming slides. So before we jump into the details of genotype, I want to cover a very basic topic that we've all studied while studying genetics, that is genes and alleles. And oftentimes there is a confusion as to what is the difference between the both. So gene is a section of a DNA which codes for a functional protein. So inside a cell, we have nucleus. Within nucleus, we have chromosomes. And if you extend a chromosome is nothing but it's tightly wound up DNA around some proteins. So if you unwind the DNA, you'll essentially get like the double helical structure, and the portion of that double helical structure is a gene, which ultimately gets transcribed and translated into a functional protein. You must have heard that we are diploid organisms, meaning that we carry two pairs of chromosomes in the nucleus of our cells. We have 46 chromosomes, 23 are from the father and 23 are from our mother. So let's consider an example of a gene. Let's say it's present on chromosome 7. So now you should have two versions of the gene, one version present on chromosome 7 derived from the father, and the other version present on chromosome 7 derived from the mother. And these two versions of the genes are nothing but alleles. So since alleles are different versions of a gene, the difference could be of a single base or segment of bases. So a genotype is basically a combination of alleles that an individual possesses. So diploid organism can either be of these three genotypes. So the first one is homozygous reference. Homozygous meaning that the bases at that location in both the versions of the gene resembles to that of the reference base, that is A, and hence it is homozygous, and since it matches to the reference, it is homozygous reference. Next, we have heterozygous alternative, as in one of the version of the gene we have a base that it matches with the base of the alternate allele, and hence it is heterozygous alternative. And lastly, we have homozygous alternative, where both the versions of the gene has a base that matches to the alternate allele at that position, and hence it is homozygous alternative. You can also see genotypes like 1/2 or 2/0, and this indicates presence of additional multiple alternate alleles. So 0 represents the reference allele, 1 represents the first alternate allele, 2 represents the second alternate allele, and so on and so forth. So let's take a look at an example. Let's look at the last two lines in this table. So looking at the variant at the 491520 position, we can see that the sample has genotype 1/2. So at that location, one of the chromosome in that sample has a base that is the first alternate allele, that is C, at that position, and the other chromosome at that position in that sample has CAC. And since CAC is the second alternate allele, it is numbered as 2, and C is the first alternate allele, and hence it's numbered as 1. Let's take a look at another example. Let's look at the last line, and again we have multiple alleles there, alternate alleles, and the genotype of the sample there is 2/0. So that position in the one of the chromosome has base G. 2 represents the second alternate allele, so that is G, and the other chromosome would have the base C, which is the reference allele, that is represented by 0 at that position. So a last part associated with the genotype is the phase. So before going to the details, let us look at these records, and here we just discussed for each of the sample, the first part of the information is the genotype, and in the previous slides we have been discussing what is genotypes and how to read these genotypes. But if you look at these genotypes closely, you will see that there are certain genotypes that are denoted by a pipe operator between the two alleles, and there are some genotypes that are represented by forward slashes. So essentially, what is the difference between them? The difference between them as is the ones that are represented by pipe operator are essentially the phased genotype, and the ones that are with the forward slashes are unphased genotype. Genotype phasing basically tells us which parental chromosome the allele belongs to. So here, for example, the first allele usually belongs to the maternal parent, and the second allele belongs to the paternal parent. So here we can see that both the alleles matches to the reference allele at that position. Let's take another example. So in this example, we can see that the maternal allele, that is the first allele, it matches the first alternate allele, that is has base G at that position, and the paternal allele has base T, which is the second alternate allele at that position. In case of unphased genotypes, we don't really know which chromosome does the allele belong to. So in this case, one of the allele has bases GTC, which matches the reference allele at that position, and the other allele matches to the first alternate allele, that is G, at that position. But we don't know whether bases GTC, the allele having bases GTC, belongs to the maternal chromosome or the paternal chromosome. So now let us look at how variations are recorded in VCF entries. So starting with SNPs. So here is an alignment between reference and the alternate allele, and you can see that at position 3 we can see a SNP, and in a VCF file the position would be 3, and the reference allele would represent the reference base, and the alternate column would represent the alternate allele base at that position. Next, taking a look at the insertion, we can see that there are certain bases inserted in the alternate allele after position 3. So in the VCF file, the alternate allele would also include a base preceding the insertion, so we could easily identify at what position is there an insertion in the alternate allele. Now taking a look at the deletion event, here we can see that there are two bases in the alternate allele at position 3 and 4 that are deleted. So while reporting a deletion in a VCF file, the alternate allele reports the base before the deletion. If there are multiple alterations, so let's say we have alternate alleles, so one of the allele has a SNP and the other allele has a deletion at position 3. This can be represented in the alternate allele column with commas representing different types of alterations that is observed at that position. In addition to these variations, there are also structural variants that can be reported in a VCF file, like a large deletion or inversion. So let's say if we have a large deletion, it can be represented in a VCF file with the position before the deletion, large deletion event happening in the reference. So in the example here, the deletion event is observed in the alternate allele from the base 101. So the position here is 100, and for the alternate allele section we see this annotation, which is described in the info with the SV type annotation, which is a deletion event, and it also provides the information on where this deletion ends. So it ends on the base 299. Inversion is another structural variation in DNA where certain sequences or certain block of sequences in a DNA sequence is inversed. So in this case, the position is reported of the reference before the inversion takes place in the alternate allele. And in the alternate allele column, we see annotation that is a structural variant type, that is inversion, and also provides the end of where the variation ends. So the end indicates that the variation continues until this position. Now that we have looked at each of the individual pieces in detail, let us put it all together and try and understand what the record means here. So the first, looking at the first record, so this variant is present on chromosome 20 at position 14370, and this variant is previously identified and classified and is a part of a database, that is dbSNP, and it has an ID RS6054257. The reference allele in the position is a G, and the variant allele has a base A at this position. This variant passes all filters, and there are 3 samples which are investigated with a total depth across all 3 samples of 14 reads, and the allele frequency of alternate allele in these sample is 0.5. Now alternate allele, to calculate alternate allele frequency, we basically calculate how many alternate alleles are observed among all alleles in the study. So since there are 3 individuals which are diploid, we have 6 chromosomes, and out of the 6 chromosomes we see 3 of them having the alternate allele at that position, and hence the alternate allele frequency for this record is 0.5. Now coming to the sample level annotations, we can see that for each sample we have information on the genotype, genotype quality, depth of the sample, and haplotype quality. So the first sample has a phased genotype, and it's a homozygous reference, since both the alleles match with the reference allele at that position, and the genotype quality is 48, and there is one read that is supporting this with haplotype quality of 51. The second sample also has a phased genotype, and it's an heterozygous alternative. We can see that the maternal allele has a base A at that position, and the paternal allele has a base G at that position. And this individual has a genotype score of 48, and it's supported by 8 reads with a haplotype quality of 51. For the last sample, we can see the genotype is unphased, and it's a homozygous alternative with a genotype quality score of 43, and there are 5 reads supporting this genotype. And since this sample is an unphased data, there are no haplotype quality scores, which are indicated by period, comma, period. Unlike VCF files, which only record variant sites, Genomic VCF contain a record for every position in the genome, regardless of whether it is a variant or a non-variant. And these genomic VCFs are generated by a GATK program called Haplotype Caller in GATK versions 3 and above. And there are two types of genomic VCFs that you can produce. So one is where there is an individual record at for every site, whether it's a variant or a non-variant. And the other type of genomic VCF would contain individual variant records for variant sites, and it's followed by non-variant sites which are grouped together into a non-variant block of records. So that's all I had for this video. I hope you found this video informative. If you did, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.